My name is Meg Keating, and I'm one of the genetic counselors here at the Greenwood Genetics Center. You are caring for a child who is currently in the hospital, such as in the neonatal intensive care unit or NICU. After evaluating your child, their provider has recommended a test called rapid whole genome sequencing. This is a genetic test that can help diagnose a large number of underlying conditions. And the results from this test may have an immediate impact on the treatment plan for your child, as well as provide information about their longer-term health and care needs. I'm here to provide you with just a little bit more information about rapid whole genome sequencing, as there are likely many different tests being ordered for your child during their hospital stay. First, let's review some background genetic concepts to better understand why this testing was recommended. Our bodies are made of millions of cells. Each cell contains a full set of our genetic instructions, which are organized into structures called chromosomes. If we stretch out the chromosomes, we begin to see the genetic code written out in letters. These letters make up thousands of individual instructions called genes. Genes code for proteins that help our bodies grow, develop, and function. If you think about the genes made up of letters providing instructions to the body, you can imagine that it is important that the instructions are written clearly in a way the body can understand. This test specifically looks at those instructions and how they're spelled. There are many different types of spelling changes that can occur within the genes that then causes problems for the way the body forms or functions. These changes are referred to as variants. Rapid whole genome sequencing is a genetic test that looks through all of our genetic information for spelling changes, not only in the pieces of genes that code for proteins called exons, but also in the information between these exons called introns. It also looks for small missing or extra pieces of DNA, known as deletions and duplications. These types of spelling changes may be called copy number variants. When this testing is ordered as rapid, the lab will expedite the analysis to achieve faster results, which is especially important when a patient is in the hospital. When possible, your provider may recommend that we get samples not just from the patient in the hospital, but also from the patient's parents. These samples will be used to help us understand a patient's test results. If both parent samples aren't available, we can still run this test for the patient alone or for the patient and just one parent. While blood should be able to be collected from the patient in the hospital, saliva swabs may be obtained from parents for testing if a blood draw is not possible. The primary reason for doing rapid whole genome sequencing is to identify a genetic cause for an individual's symptoms in a very short time frame. Alternatively, this test can help to rule out some possible genetic causes. Both outcomes have the potential to directly impact a patient's care while they are hospitalized. This test is phenotype driven, meaning that your provider will share the patient's clinical features with the lab to help them filter through the genetic data in an informative way. Our intention is not to report all genetic changes an individual has, but rather those that are relevant to their medical history and can help drive treatment and management decisions. These changes are considered primary findings. There are three different results that we can get from this testing. The first is a negative or normal result. This means that we looked across the entire genome and didn't find any changes that we think are causing problems. Negative results can still provide important information to a patient's doctors and may still inform changes to a patient's care plan or help determine further testing options. We can also find a positive or abnormal result. This means that we found a change in a gene that we think is causing part or all of a patient's symptoms. Finding a genetic cause can also tell us if there are any other related health concerns we can prepare for and potentially provide treatment options specific to the patient's genetic diagnosis. The third result is a variant of uncertain significance, or VUS. This means we found a change in a gene, but we aren't quite sure if the spelling change results in any health concerns or if it is just a part of normal variation that we all have. In these situations, the patient's provider might recommend additional testing or evaluations, or they might wait until we've learned more information about the change. As we learn more information, these variants of uncertain significance can be either upgraded to pathogenic or disease causing, or downgraded to negative or normal results. Please know that in genetics, we are learning new things all the time. So even if you receive a normal result now, it is possible that we will contact you in the future with an update. We understand the urgency and importance of completing this testing while a patient is in the hospital. Preliminary results of this test will be available within 5 to 7 days after samples are received by our lab. These preliminary results will include any genetic changes that immediately appear to be a possible answer. If there is no clear answer at the time preliminary results are released, providers are encouraged to provide updates about any symptoms that may have developed after testing was initially ordered. A final report will be delivered by 14 days after the lab receives the sample. Your child's provider in the hospital will inform you of results from this testing. If results come back abnormal or uncertain, a GGC provider may contact you to set up a time to review results in more detail and discuss next steps. The intention of this testing is to identify a cause for a patient's concerns. However, we can sometimes uncover incidental or unexpected information that can impact the patient and possibly their family members. For example, the test may reveal that a patient may develop additional health problems in the future. If parent samples are included, it may reveal that a patient's parent has the same genetic condition as their child, that parents are related to each other, or even that one of a patient's parents is not their biological parent. Although rapid whole genome sequencing is our most comprehensive genetic test offered now, it is also important to keep in mind that this test cannot detect all possible genetic changes. In some cases where rapid genome sequencing is negative, further testing may be recommended. A genetics provider at the Greenwood Genetics Center will contact you with any further recommendations. If you have any questions about this testing, reach out to your Greenwood Genetics Center providers and we'll be happy to answer your questions.